Okay, good morning students. See here in the chapter protein and amino acid metabolism. So already in the previous classes we have seen how the amino acids and proteins have been classified and like structural organization of proteins, like how they have been organized into primary, secondary, tertiary and quaternary structure. Then today in this class I am going to explain you digestion and absorption of the protein. And you know that digestion, it's a process of the breaking of the complex molecules into simpler substances and preferably water soluble, which can be easily digested by the small intestine. Okay, see here, digestion means it's a process where the organic molecules of the foodstuffs are converted, okay, or complex ones are converted into simpler ones, and that will be preferably water soluble. Those can be easily digested by the small intestine. Now here the if you see that mainly digestion takes place in the mouth. In the mouth during the chewing of the chewing of the food, mastication, it mixes with the saliva, and then food bolus through esophagus, it will be passed through the stomach. There it mixes with the concentrated gastric juice. The contents of the gastric juice it contains gastric enzymes and HCl. And then the acidic food bolus, then it will be released into the duodenum. There it exposes to the pancreas, which also contain pancreatic enzymes, and that is bicarbonate in nature. And here it also mixes with the bile. Okay, and the final digestion takes place in the small intestine. So let us discuss about the digestion of the proteins in mouth. What happens to the proteins once you ingest it in the mouth, stomach, and then pancreas, final digestion in the small intestine. Now here coming to this one, already you know that saliva, here it is secreted by salivary glands, and it contains 90%, almost 99.5% water is there, and here mucin and salivary enzymes are there. And gastric juice, its composition is 99% of the water is there, and 0.2 to 0.5% of the HCl is there. As the acid is there in the gastric juice, its pH is less than 2, and digestive enzymes are present like renin, pepsin and this one. And even the bile is produced, liver produces bile, and bile contains bile salts which help in the emulsification and neutralization of the acid secretion. Okay, see here lipids mainly digested. See here this bile which is secreted from the liver, which plays an important role in the digestion of the fats. Now here the pancreatic juice, which is non-viscid watery fluid, and it mainly contains, if you see the composition of the pancreatic juice, it contains proteins, organic and inorganic compounds, and it contains many proteolytic enzymes. They are trypsin, chymotrypsin, elastase, carboxypeptidase, amylase, lipase, phospholipases, cholesterol esterases, ribonuclease. These are the enzymes which are present in the pancreatic juice. Anyway, pancreatic juice contains like alkaline in nature, which neutralizes the acidity of the food. And final digestion in the intestine. Here the intestinal mucosal cell secretes intestinal juice, and here which mainly contains the enzymes like aminopeptidases, disaccharidases, oligosaccharidases. These are the enzymes. Okay, now here coming to the digestion of the proteins. Okay, see here already you know that in the previous classes, protein digestion, which involves the the proteins are the macromolecules with definite size and shape, and proteins are made up of amino acids. These amino acids are joined by peptide bonds. So digestion of the protein involves breakdown of the peptide bonds and to release the amino acids. So the end products of proteins are amino acids. So amino acids which are joined each other by peptide bond in the formation of the protein. During the digestion process, those peptide bonds has been hydrolyzed by various enzymes which are present in the mouth, like stomach, pancreas and in the intestine. They will hydrolyze the peptide bonds and release the free amino acid. Okay, so and here the daily dietary protein load for digestion is around nearly 100 to 200 grams. Okay, nearly 100 to 200 grams of the proteins has to be digested in your body in a day. Out of that, those proteins which are subjected to digestion may be obtained from two sources. dietary sources and endogenous sources. Dietary sources means exogenous, that what we are taking, the proteins what we are taking in the diet. Nearly we will take 50 to 100 grams of proteins in a day. And endogenous proteins means by the translation process, even proteins can synthesize in your body. So those proteins which are synthesized, which have already synthesized in your body, are called endogenous proteins. Their concentration is 30 to 100 grams per day. Okay, so what you have to understand, digestion of the proteins is defined as the breakdown of the complex materials, that is proteins, into simpler amino acids right here. And proteins which are subjected to digestion are obtained from two sources: dietary sources, that is nearly 50 to 100 grams per day, and endogenous proteins, 30 to 100 grams per day. Okay, these proteins, whether it is endo or exogenous protein, it need to undergo digestion. So proteins are, I already mentioned, proteins are digested by breaking the peptide bonds. So in the body there is a group of enzymes, in the biological fluids there is a group of enzymes are there which will cleave the which will break the peptide bonds. Those enzymes are called peptidases. Okay, see here, the peptidases are the enzymes. Okay, name of the enzyme, peptidases are the enzymes which are specific in hydrolyzing or breaking the peptide bonds, and those peptidases enzymes which comes under hydrolases enzymes. Okay, now here there are two types of endopeptidases. There are two types of peptidase enzymes are available in our biological fluids. So what is the action of the peptidase? Peptidase is an enzyme which will break the peptide bonds which are present among the amino acids of the proteins. So in your body during the digestion process, peptidase enzymes plays an important role. There are two types of peptidases are there: endopeptidase and exopeptidase. Okay, what do you mean by endopeptidase? The name only indicates these type of enzymes catalyze the internal peptide bonds and convert the large fragment of the peptides into small fragments. Okay, examples of the endopeptidases are pepsin and trypsin. Then the next one is exopeptidase. Exo means these will catalyze the peptide bonds either from the C terminal or from the N terminal. If you remember the structure of the protein, what will be there in the structure of the protein? In the primary structure, sequence of the amino acids will be there. One side there is free carboxyl group is there, that is called C terminal. Another side there is N terminal is there, where amino group is there, free amino group, the terminal is called N terminal. Okay, now these endopeptidase enzymes which will catalyze the internal peptide bonds, not from the ends. They will randomly cleave the internal peptide bonds and convert a large fragment of the protein into small fragments. Examples of this endopeptidases are pepsin and trypsin. Then coming to the exopeptidases, these are catalyze the peptide bonds either from the C terminal or from the N terminal. So there are two examples are there, two enzymes are there under exopeptidase: carboxypeptidases and aminopeptidases. So what do you mean by carboxypeptidase? Means this enzyme catalyze the peptide bond from the C terminal. Aminopeptidase means catalyze the peptide bond from the amino terminal. And now coming to the digestion, and one more thing is here, all the endopeptidases or exopeptidases, they are present as inactive forms. Okay, so always they will be secreted in the body and they will be stored in the body in the form of inactive forms. Whenever the need is there for those enzymes, means when the digestion process is taking place, then only they will be converted to active forms. Other than the remaining times, they will be remaining as the inactive forms. Why they have to secrete as inactive forms? Means because all the tissues in your body made up of proteins only. If they are remain active all the time, what will happen? They will break its own protein. Okay, so to prevent the self digestion, auto digestion, these peptidase enzymes are secreted as inactive forms. Whenever its need is there, then only they will be converted to active form. Now here coming to the protein digestion, first in the mouth, there is no digestion of the proteins in the mouth, since there is no peptidase enzymes in your saliva. Okay, so that means proteins will not undergo any change in the mouth. Then the next one is digestion in the stomach. Once the food enter into the stomach, it mixes with the HCl. Okay, gastric juice which contains HCl and an enzyme that is pepsinogen. So HCl is the pepsinogen is the proenzyme. So that is the inactive form. Now here HCl, which functions as it denatures the protein and it kills the microorganisms. Right here HCl, what is the action of the HCl? The role of HCl in protein digestion is it denatures the protein. Proteins, it's a property of the protein is when exposed to when it is treated with the acid, they will undergo denaturation. Right here, denaturation means only the primary structure will not undergo any change, but changes will be seen in the secondary, tertiary and quaternary structure of a protein. And not only that, even the HCl, it kills the microorganisms. Now coming to the next one is pepsin. Actually pepsinogen is in active form. It is secreted by the chief cells of the stomach, and here whenever the HCl which is there in the stomach, that will convert the inactive pepsinogen to active pepsin. Right, some amount of the pepsin is released into the stomach. The rest of the pepsin itself convert the pepsinogen to pepsin. This process is known as autocatalysis. I hope you are understanding. First we have taken food, protein in the mouth. There is no digestion in the mouth. In the stomach, the proteins are mixed with the HCl which denatures the protein and improves the digestibility of the protein. The second one is it mixes with the enzyme pepsinogen. Okay, pepsinogen I mentioned, it is an inactive form. So pepsinogen initially with the help of HCl, it is converted to pepsin. Pepsin is the active form, pepsinogen is an inactive form. Once pepsin is released some amount, that in turn converts the rest of the pepsinogen into pepsin. So this process is known as autocatalysis. Now what is the action of the enzyme pepsin? Pepsin is an endopeptidase right here. So it will catalyze the internal peptide bonds and convert proteins into peptides. Okay, small fragments. Those small fragments we can call it as peptides or peptones. So pepsin which is there in the stomach, that will cleave the proteins up to peptides and peptones. Now this pepsin is acid stable because the pH of the stomach is less than 2 because of the presence of HCl. But this enzyme pepsin is acid stable. It is active low pH below 2. Okay, and here now I already mentioned pepsin is an endopeptidase, catalyzes the internal peptide bonds, and dietary proteins are hydrolyzed by peptine up to proteoses and peptines. Now even in the infant stomach, along with the HCl and pepsin, there is an enzyme is there that is called renin. Okay, renin is active only in the infants and children. What is the action of the renin means here it converts the milk protein, that is casein. Okay, please make note down. Casein is a milk protein. So that casein is acted upon by the renin which is there in the stomach to calcium paracaseinate. Okay, casein is the milk protein. Renin is an enzyme, proteolytic enzyme which is active in the stomach of the infants and children. What is the action of this renin? It converts a milk protein casein into calcium paracaseinate. Okay, and this calcium paracaseinate is further acted upon by the casein. So it is involved in the curdling of the milk. Okay, renin is an enzyme which involved in the curdling of the milk, and renin is absent in the adults. Okay, because up to 6 months of the life, first 6 months, the baby depends completely on the milk. So milk protein casein can be easily digested in the infants by this renin. Okay, after that, once we start in the weaning or solid foods, here the renin will become inactive. So renin is absent in adults. Then coming to the next one, digestion of the proteins in the pancreatic juice. Okay, pancreatic juice contains bicarbonates, trypsin, chymotrypsin, elastase and carboxypeptidase. Now here in the acid, the food in the stomach becomes acidic. So once the food bolus enters into the duodenum, there it mixes with the pancreatic juice. So pancreatic juice, as it contains sodium bicarbonate, first it neutralizes the acidity of the food bolus. Then the proteins are then acted upon by the various enzymes which are present in the pancreatic juice, or proteolytic enzymes or trypsin, chymotrypsin, elastase and carboxypeptidase. Now here how they will be secreted, these four enzymes means the entry of the food into the pancreas release the two hormones, that is secretin and cholecystokinin. Okay, and this cholecystokinin which will these enzymes are secreted. Cholecystokinin stimulates the pancreatic release of the pancreatic enzymes. But please make a note here, all the pancreatic enzymes also they are secreted as the inactive forms. Okay, all the enzymes secreted as inactive forms, and how these inactive forms will be converted to active form means by the enzyme enterokinase. I already mentioned all the peptidase enzymes which are present in the in the stomach or in the pancreas intestinal juice. First they will be secreted as the inactive forms. Whenever a special stimulus is there, need is there, then only they will be converted to active forms. In the stomach, how the pepsinogen is converted initially by the HCl, later on by the pepsin itself. Even here in the pancreatic juice also many proteolytic enzymes are there, but they will be secreted as the inactive forms like trypsinogen, chymotrypsinogen, procarboxypeptidase, proelastase, procolipase. All the left ones are the inactive forms. By the appropriate stimulus only they will be converted to the active forms. Now here initially the enzyme enteropeptidase which will convert trypsinogen inactive form to active trypsin. Once some amount of the trypsin is released, that will convert the remaining inactive forms into active forms. Okay, trypsin then convert chymotrypsinogen to chymotrypsin, procarboxypeptidase inactive form to active carboxypeptidase A, B, aminopeptidases, and even this trypsin will convert proelastase inactive form to active elastase, and even it convert procolipase to colipase. Like that, initially inactive form is trypsinogen is being converted to active by the enteropeptidase. The released trypsin in turn convert the rest of the enzymes inactive forms to active forms. So once all these enzymes are gets activated, trypsin, chymotrypsin, carboxypeptidase, elastase and colipase, they will act on the peptides and proteins, because already proteins have been digested up to peptones and peptides were in the stomach. Now here in the pancreas, those proteins and peptides have been acted upon by all these enzymes and converted up to tripeptides, dipeptides, and few free amino acids are released. Then coming to the next digestion, final digestion in the intestinal juice, where the entry of the food into the small intestine release intestinal juice which contain two enzymes, aminopeptidases and dipeptidases. Okay, aminopeptidase means it is an exopeptidase which will remove the one amino acid from the N-terminal, and dipeptidases means they will act on the dipeptides, because the which type of protein which enter into the intestinal juice, already in the pancreas only proteins have been broken up to dipeptides, tripeptides. Okay, those dipeptides, two amino acids, three peptides have been acted upon by intestinal enzymes dipeptidase, that will break the bond, peptide bond, and releases the amino acid. So like that, with the help of all the enzymes of the stomach and the pancreas and the intestine, the proteins have been completely break down up to amino acids. Okay, so in the intestinal human there won't be any proteins. By that time already they will be broken up to amino acids. Okay, see here, this is the proteolytic enzymes and their action. First, there is no digestion in the mouth. In the stomach, which enzyme? Pepsin enzyme is there, that will convert the protein into small peptides. Then coming to the pancreas, there are four enzymes are there: trypsin, chymotrypsin, carboxypeptidase and elastase. What is the action of the trypsin? It specially acts on the basic amino acids of arginine, lysine and histidine. Okay, and this trypsin activates the trypsinogen to trypsin, and it also converts all the rest of the inactive forms to active forms. Now here carboxypeptidase and elastase, they will act upon the peptide bonds from the carboxy terminal. Then coming to the final digestion in the small intestine, there are two enzymes will be there: aminopeptidase and dipeptidase. Aminopeptidase act upon the amino terminal amino acids. Dipeptidase acts on the amino acid and releases the free amino acids. So that's it about the digestion of the proteins. Proteins at the end of what are the end products of digestion of the proteins? They will be converted up to amino acids. Okay, now we are coming to the next process. Once the amino acid digestion is completed, which is followed by the absorption for further utilization. Absorption is nothing but where are your amino acids right now? They are in the intestinal lumen. Then they have to cross the intestinal mucosal cell to enter into the blood circulation for further utilization. This transport of the amino acids from the intestinal lumen into the blood circulation, the process is known as absorption. In the absorption, various transport mechanisms are involved in the transport of amino acids from the intestinal mucosal cell into the blood circulation. Right here, and first amino acids are transported from the lumen to the intestinal mucosal cell, and from the mucosal cell they will be transported into the blood circulation. Okay, so in order to transport the amino acids from the lumen to the mucosal cell, there are different transporters are required. So those transporters are sodium dependent transporter, or few transporters are there, okay, which will transport exclusively neutral amino acids, and few transporters will transport basic amino acids, and a special transporter is required for the transport of beta amino acid, and for amino these acidic amino acids. So that means there are different transporters should be available in the intestinal mucosal membrane for the transport of various amino acids. Okay, so these are the how, let us see this one. This picture will explain you how the amino acids has been transported from intestinal lumen to the portal circulation. If you remember the absorption of the glucose, it is exactly same as like glucose, only in place of glucose you have to write about the amino acid. Okay, right here what is the how it will be transported. Now your amino acid will be there in the intestinal lumen. How it will be entered into the portal circulation means, first here this amino acid has to be transported to the cytosol, mucosal cell membrane mucosal cell. So how means they will transport the sodium dependent glucose transporter, okay, from here to here. From here to here it is transported by active transport. What do you mean by active transport? Active transport means here the solute moves down the concentration, against the concentration gradient, means from low concentration to higher concentration, and it requires energy, and it requires a special carrier, a special transporter is required. So this amino acid from the lumen to the cell, okay, to the intestinal mucosal cell, they will be transported by active transport. The transporter, name of the transporter here it is sodium dependent transport. Okay, here means the green one is the transporter which has two sites, okay, for the binding of amino acid and sodium. So as the concentration of the sodium is high in the lumen when compared to cell, the sodium moves down the concentration gradient. Okay, when it is moving from here to here, it will drag one molecule of amino acid from the lumen to the cytosol. Now here the sodium work is done, okay, and in order to maintain the cell neutrality, the sodium should come out of the cell and enters into the lumen. So that process will take place in exchange of the potassium, that is called sodium potassium pump. Okay, see here, first amino acid which is there in the lumen has to enter into the mucosal cell. In order to enter into that one, it has to cross the membrane. Membrane contains a special transporter that will help in the transport of the amino acid. Already I mentioned those transporters are there, special transporters are there for basic amino acids, acidic amino acids, neutral amino acids, etc. Now that transporter is the sodium dependent. The concentration of the sodium is high in the lumen when compared to the cytosol. Sodium moves down the concentration gradient and it drags the one molecule of amino acid from the lumen to the cytosol. In order to maintain the neutrality of the cell, sodium will come out of the cell in exchange of the potassium. So this process is done by sodium potassium ATPase. So that means this exchange process needs energy that is provided by hydrolysis of the ATP. This reaction is catalyzed by the enzyme sodium potassium ATPase, that we can call it as sodium potassium pump. So how many sodium it will pump means, this sodium potassium pump, it will pump 3 sodiums from cytosol to the circulation in exchange of the 2 potassium. Now here your amino acid is there in the cytosol, means in the mucosal cell, that will enter into the portal circulation by passive transport. Okay, that is passive diffusion. So like that, amino acids, whatever is there in the, these are the end products. End products are there in the lumen. By active transport, they will be transported to the mucosal cell. From mucosal cell to the portal circulation, they will be transported by facilitated diffusion. And even there is a MEISTERS cycle is there, that will explains you transport of amino acids across the cell membrane. Okay, and that is called gamma glutamyl cycle. That I think this is not necessary for you. And the next one is absorption of the intact proteins and polypeptides. Okay, sometimes immediately after birth, the small intestine of the infants can absorb the intact proteins and polypeptides. Okay, so that means the proteins which are there immediately after birth, directly and not amino acids, proteins can be absorbed from the small intestine. Okay. The uptake of the proteins, this process occurs by endocytosis or pinocytosis. So the direct absorption of the intact protein is very important in the early life for the transfer of maternal immunoglobulin to the offspring. Okay, I think you are following. Immediately soon after the birth, directly in adults only amino acids will be absorbed, but in the stomach of the infants, okay, in the small intestine of the infants, directly proteins can be absorbed by exocytosis or endocytosis. Why? Because in order to transfer the maternal, means immunoglobulins, which gives immunity from the mother to the baby. The intact proteins and polypeptides are not absorbed by the adult intestine. Okay, only intact proteins are absorbed in the early life, soon after birth, but in the adults there won't be any absorption of the proteins. Okay, then finally coming to the absorption of the abnormalities which will be associated with the protein absorption is Hartnupp's disease. And this is due to the defect in the absorption, that is transport of the aromatic amino acids from small intestine as well as from the renal tubules. Okay, here what happens here? There is a defect in the aromatic amino acids. Name the aromatic amino acids: phenylalanine, tyrosine and tryptophan. Their absorption is defective either from the small intestine or from the renal tubules. So because of this why it is called Hartnupp's disease means that disease symptoms can be first observed in Hartnupp's family. Hence the disease is named as Hartnupp's disease. Here the patient may suffer from pellagra-like symptoms, and it can be high protein diet with the niacin. It can be treated. Okay, and there in the Hartnupp's disease there is decrease in the absorption of the neutral amino acids like tryptophan, alanine, serine and valine. So tryptophan is affected. Okay, that's it about the protein digestion and absorption. It is an essay question for you. Okay, in the next coming class, now your amino acids are there. You have taken protein in the diet. By the time it will be reaching the small intestine, they will be converted to amino acid. Now those amino acids, now by the various transport mechanisms, they will enter into the blood circulation. Now in the next coming class, I will explain you what is the fate of that amino acid once it enter into the blood circulation. Thank you.